Now, what if you need a little more? We have what are referred to as endogenous pyrogens. These are going to be cytokines, chemokines that will be activated and then will cause fever. TNF alpha, IL-1 beta, IL-6 will react, cause the hypothalamus to react, which will increase overall temperature for the body. You get localized, you definitely start with a localized. If it's an infection, a wound infection, you will get localized elevated temperatures. If enough TNF alpha, IL-1 beta, IL-6 gets into the bloodstream, then you can end with a body-wide temperature increase. Do you ever wonder why a wound starts to feel warm? It's not just the collection of the fluids in that area, but it's also the pyrogens. There are also outside exogenous pyrogens. LPS, if it gets into the bloodstream, gets into the tissues, will cause cells to increase their metabolism, start to burn through their energy savings. ATP, all this, increase their metabolism. And you got to remember, thanks to the second law of thermodynamics, anytime you change things, change energy from one form to another, there's going to be some loss. We call that loss heat. So you're going to heat up a certain area. Gets into the bloodstream, LPS gets in the blood, goes to the hypothalamus, just like TNF alpha, IL-1 beta, will cause you to have a system-wide fever. Now, once things progress, once things get to a certain point, we see that the TNF alpha, IL-1 beta, IL-6 will trigger what is known as the acute phase response. It's a response that will be dependent upon which tissues you're talking about. IL-1 beta, IL-6, TNF alpha in the liver will cause what are referred to as the acute phase proteins, the C-reactive proteins, mannose binding lectins. These will trigger activation of complement, opsonization. So, an activated macrophage in the liver triggers an uptick of complement opsonization. In the bone marrow, you get IL-1 beta, IL-6, TNF alpha, causes neutrophils to mobilize. They will go hunt down anything not you in the bone marrow. Hypothalamus, increase blood temperature, body temperature, which will decrease viral replication, bacterial replication. Turns out bacteria only like a certain range of body temperatures. Those that infect us at 37 degrees Celsius, 98.6 degrees Fahrenheit, that's usually at the top end of where they like to be. Generally, they like to be down more around 35. So you get a fever, you're bumping it up to 38, 39, you're now getting out of their comfort zone. They will not do as well. Fat muscle, again, it's going to tell those cells in the fat cells, the muscle cells, to start mobilizing all their energy, increase flow, increase body temperature. This may be localized or it may be system-wide. Again, this is going to interfere with viral replication and bacterial replication. What you see is that IL-6 released into the liver by, say, an activated macrophage or some cell within the liver that's on patrol, comes across something not you. It will act on what are referred to as hepatocytes. Hepato means liver, so these are going to be granulocytes that are liver only. They are then going to start overproducing mannose binding lectins, fibrinogen, C-reactive proteins, serum amyloid proteins. The C-reactive proteins, serum amyloid proteins, have been shown to bind to bacterial surfaces They bind the phosphocholine that's found on bacterial surfaces, may be found on some of our surfaces, but if the liver's producing it and producing it in high amounts, it means that there must be an active infection anywhere, so the body will risk cross-reactivity. The increase in mannose binding lectins is not just to increase the chances of complement cascade activation, but to act as opsonizers. Opsonizes, starts punching holes in the bacteria, also aids in the grabbing hold by macrophages, neutrophils, and their phagocytosis and digestion. Interferon-producing cells, also known as IPCs, when they come into contact with viruses or virus-infected cells, will start to the overproduction of IFN alpha, IFN beta. These cells, when they overproduce IFN alpha, IFN beta, interferon alpha, interferon beta, will induce other cells to start making more antiviral proteins. They will start going and linking adenosine. They will start activating certain kinases inside the cells. Increase MHC class 1 expression to increase the chances of a viral epitope being expressed on the surface to be seen by an activated CD8 cytotoxic T cell. Interferon will also activate any neighboring or passing by NK cells, the natural killer cells, that will also go and hunt, trying to find any cell expressing or showing signs of viral infection. Difference here between the MHC class 1, the going for the cytotoxic T cells, and the activating NK cells. NK cells are indiscriminate. If a cell is showing any signs of viral protein on its surface, even if it's just going to, be, a viral spike, or the infected shell is showing signs of distress, natural killer cell will kill it irregardless. Sometimes the NK cells have been shown to kill uninfected cells. Better to lose the cell than lose the tissue. Cytotoxic T cells are highly specific. They will only go after a specific cell that is showing signs of a specific viral infection. Specificity. That's the difference between the T cells and the natural killer cells. Interferon alpha beta will also act as chemokines to recruit in lymphocytes. Bring in the B cells, the T cells. Get them where they need to be. So you look at viral infection, day 1 through day 10. Low titer on the virus, as we're seeing here, that increases within the first and then starts to decrease within the first week, nearly gone by day 10. Starting end of day 1 into day 2, you see the production of interferon alpha beta, TNF, IL-12. These are known as antivirals. The antivirals will kickstart innate cellular response. NK cells come in, as well as dendritic cells will come in and will start clearing out. NK cells will kill, induce apoptosis, directly kill. Macrophages, dendritic cells will look for viruses that are outside the cell, that are going from cell host to cell host. As the interferon production starts to decrease, natural killer cells, they start to decrease, the activated ones. We see the cell mediated. The adaptive response kicks in. As the adaptive response kicks in, virus titer goes down, because the T cells are definitely going to hunt down and kill all viral infected cells. The interferons, the natural killer cells, will slow down the spread, but it's actually the T cell that's going to remove the infection. So what we see when we look at this from the NK cell and the T cell responses, the NK cell depends upon a host of signals, a balancing of signals. NK cells can recognize, to an extent, MHC class 1. But under normal cell, as we see here, it's receiving a negative. What was there is nothing that is not viral in origin. This is natural, normal, human, me proteins, epitopes. So no signal, co-signal, natural killer cell continues on. But if the MHC class 1 doesn't interact because the cell is infected with a virus that is inhibiting MHC class 1 expression, or it's interfering with somehow with something being loaded into the cleft of MHC class 1, as that's showing here in the figure. So the altered M8 or absent MHC class 1 does not give the initial no, but the receptor still is able to bind. The natural killer cell's receptor still binds. This will trigger the natural killer cell to disgorge its granules onto the surface of this potentially infected cell. Potentially. The cell itself could be damaged due to, a wound or whatnot. Could reach the end of its age. So it doesn't mean this has to have been viral infected. It could just be damaged, which still allows the NK cell to kill it. Now, one of the things we talked about in an earlier lecture in this chapter was that sometimes the receptors on these cells are going to be highly specific or not so much. We see that when you look at the receptors, the genes responsible for the receptors on natural killer cells. Whole host of genes spread across different chromosomes, which give us the leukocyte receptor complex, or the natural killer complex. We get numerous genes that will come up with the different conserved domains that will all come together to form a complete complex that we call the receptor. The different genes, the different parts are going to be found on different chromosomes, as they're seeing here, chromosome 19, chromosome 12. When it comes to the receptors, the natural killer cell receptors, we see that there's going to be activating and inhibitory receptors. Turns out the natural killer cells are always on the verge of killing. Okay? CD8 cells, they are quiescent, just hanging out in the lymph node until they're activated, then they go hunt, kill, and then they go back to the lymph node, done. NK cells are constantly on patrol, always on the verge of killing something. Well, how are they always on the verge? Why aren't they always killing? Because it's a balancing act, activating and inhibiting signals. Push one way over the other, the cell will start to kill. It will kill whatever is in its vicinity. You see you have ICAM type receptors, and CD94, composed of NKG, natural killer, G2, C, and E. These are the activating and then the inhibiting. ICAM-like, ICAM domain-like, and a CD94-like. Which ones get more of a signal at any given time will determine whether or not the natural killer cell kills or is inhibited from killing and just moves on. Now, this is so important that the natural killer cells have set receptors on their surface that look just for things not you. The NKG2D, which are a set of natural killer cells, receptors. These are cytotoxicity receptors that will then look for and interact with domains of immunological molecules, domains of FC receptors. The receptors for certain molecules you see here are MHC-like. The ligands for these guys that are going to be on the surface of the target are MHC-like. They may not be MHC themselves, but as you can see, they have domains and cytoplasmic tails that are quite similar to.